Medical Cell Biophysics is a department of the MIRA Institute for Biomedical Technology and Technical Medicine at the University of Twente. It develops pioneering, user-friendly technologies that detect illnesses faster, more sensitively and earlier in the disease cycle. It is an incubator for several startup companies as well.
In our Medical Cell Biophysics group, we are developing technology for new diagnostics, and we do that for cancer, and we also do it for very simple applications like AIDS or point-of-care analysis.
For instance, cell count, that uses magnetism to count circulating tumor cells to find out if a treatment is effective. Leon ter Stappen won the prestigious American Prix Galien with this technology.
CellSearch is the first system ever on the market which can count tumor cells in blood. And by doing so, you can actually monitor patients which are diagnosed with metastatic breast, prostate or colon cancer. We use very small magnetic particles labeled with antibodies, which are specific for epithelial cells. Epithelial cells are the basis of carcinoma cells, cancer cells. These are conjugated, we add them to the blood automatically by an instrument. The beads and the antibodies recognize the tumor cells. We use magnets to then enrich them out of the blood. Then we stain them with fluorescently labeled antibodies so that the cells which we captured are recognized. And then we use an automated fluorescent microscope to count the number of cells in the 7.5 mL of blood. At the diagnosis, you determine whether there are tumor cells in blood. And then after 3 to 4 weeks of treatment, when the tumor cells are not disappeared, the treatment is not working. You should either stop the treatment or you should switch to an alternative treatment. The big advantage of CellSearch is that you also can obtain the cells, so you can actually investigate whether the targets of your treatment is on the cell. So that would mean that you can do that in addition, and that would mean that you can immediately say this patient needs that treatment.
Now, ter Stappen's group is leading a large new European research project called CT Cell Trap. This new technology will find all tumor cells circulating in all of the patient's blood.
So in principle you can analyze 1 to 5 liters of blood in 30 minutes to an hour. You get all the cells, and that means that you not only can tell how the patients will be doing, you can actually guide the individual treatment of each patient by investigating to which treatment does this tumor cell react.
Specifically for poor African countries, Medical Cell Biophysics develops a new, inexpensive device for effective monitoring of AIDS.
There are no tools, there are no experienced people, so what we are trying to do is to develop a test to monitor the disease of AIDS, where you just can simply take a blood stick and you immediately get an answer, whether you should treat or not treat.
The current methods are use big machines. The tests are carried out by qualified, well-trained personnel. Our test would be very simple and could be done with a drop of blood by untrained persons, so that would help mainly the people in the rural areas of Africa.
At Medical Cell Biophysics, Kees Otto developed a unique method to investigate cell processes leading to disease at the very small molecular level. Otto makes this technology widely available through the spin-off company HybriScan.
We develop here extremely sensitive microscopic techniques to look at living cells. We are at the front of this research field worldwide, where we can detect individual molecules inside cells with fluorescent spectroscopy and big clusters of molecules with Raman spectroscopy. And by correlating these, we can learn about the development of diseases inside cells.
In their daily routine, doctors need reliable diagnostics for diseases like meningitis and leukemia. For that purpose, Medical Cell Biophysics designed a new technology that will be commercialized through a spin-off. Both the company and the product are called ViCap.
ViCap is developing a point-of-care cartridge for the analysis of spinal fluid for the reason to detect meningitis and for staging leukemia in patients with leukemia. The cartridge goes into this microscope. The microscope acquires an image from the sieve. The next step is to perform image analysis to count the number of cells on the sieve.
The Department of Medical Cell Biophysics conducts new nano-research as well. The project NanoLock looks into the DNA of individual tumor cells.
This is the microfluidic chip which is in there, and you see an amplification there on the screen. And there you see the individual wires, basically channels, where individual cells can be moved and can be treated and can be manipulated. The ultimate goal is to actually help patients which are treated for cancer. So at the moment you do not always know what treatment to give. What we have found in our work on circulating tumor cells is that they are very heterogeneous, so they are different. And when you start treatment, they often mutate and differentiate and they change. So what we really need is a technology we can rapidly look at the individual tumor cells. And that's what this system actually is doing. So when the tumor is changing, we can actually literally see this with this technique and then adapt our treatment accordingly.
Finally, the department has witnessed the development of the IBIS MX96, a groundbreaking combination of microfluidics, camera, computer and biosensor technology. It can detect all diseases that cause changes in the composition of human bodily fluids, using just a single droplet of blood.
Normally when there is a disease, then the composition of blood will change. For instance, if you have rheumatoid arthritis, then this is a really complex disease and many components will change. This instrument, the IBIS MX96, enables you to measure the composition of blood very fast in a sensitive way, and it can monitor, for instance, 96 tests instantly. Yeah, this technology works label-free. It means you don't have to add anything to the blood, like for instance radioactivity. And in this way you can get your results cheaper and faster. We sold already these instruments to pharmaceutical companies because it enables them to speed up the development of medicines.
The first IBIS milestone is a biosensor chip for diagnostics of rheumatoid arthritis. But more chips will be developed for more diseases, including cancer, and for the purpose of blood typing as well. By developing and commercializing these technologies for patient care, Medical Cell Biophysics contributes to the mission of the MIRA Institute of the University of Twente.